Good afternoon to all of you. I, Dr. Preeti Singh, the convener of this national webinar, on behalf of the Department of Botany, SS Khanna Girls PG College, Prayagraj, cordially welcome our chairperson, Dr. Asha Seth, our principal, Professor Lalma Singh, and the resource persons, Dr. Ravi London from the School of Biotechnology, Jawaharlal Nehru University, New Delhi, and Dr. Abhishek Kumar from the Institute of Bioinformatics, Bangalore. I also welcome the participants who have joined the webinar. The purpose of organizing this webinar is to remove misconceptions and spread awareness among the people about the COVID-19 pandemic. Now, I request our principal, Professor Lalima Singh, to say a few words about this webinar. Ma'am, kindly join us.
Yes. You can hear me, Preeti?
Yeah.
Should I start?
Ma'am.
Okay. Good afternoon and good evening to one and all present in this national webinar organized by the Department of Botany of SS Khanna Girls Degree College. Today, as the entire world finds itself battling with the challenge of COVID-19 pandemic, the words of Annie Bradstreet come to my mind, who said, if we had no winter, the spring would not be so pleasant. And if we did not sometime taste adversity, prosperity would be not welcomed. The present situation that seems so difficult and adverse to us, in my opinion, should be taken as an opportunity to learn better, to make better choices, to adopt a better lifestyle, and we should be compassionate towards all. I would like to welcome the speakers of the day, Dr. Ravi London and Dr. Abhishek, who have been kind enough to grace this seminar and enrich the participants with their knowledge. The first speaker of this webinar, Dr. Ravi London. Dr. Ravi London, who is currently working as assistant professor at the School of Biotechnology, Jawaharlal Nehru University. He has expertise in viral immunology and infectious disease biology. After completing his PhD from the University of Zurich, Switzerland, and then postdoctoral research from University of California, San Francisco, and University of Hawaii, where he was also a staff scientist, Dr. Tandon established his Laboratory of AIDS Research and Immunology at the School of Biotechnology, JNU, in 2014. He published several research articles and obtained many research grants from various funding agencies. We also have amongst us Dr. Abhishek Kumar, who is an eminent computational biologist working in the Institute of Bioinformatics as faculty research scientist. His laboratory is focusing on several biological and biomedical problems to resolve using genomic and bioinformatic methods. Dr. Abhishek, too, did his PhD from Bielefeld, Germany, and had been a different scientific and has held different scientific positions as postdoctoral in Germany and as well as in Italy. He has very impressive publication records in international collaboration. We are proud to have such eminent scholars with us in this national webinar. You all are dynamic personalities and you have achieved so many accolades. It's a moment of pride for Khanna Girls Degree College. You both scholars of repute are really inspiring for these young generation, the young scientists who are joining this webinar. I welcome you all on behalf of SS Khanna Girls Degree College and also extend my gratitude to you for being a part of this day and sharing your knowledge with all of us. Thank you. Thank you so much.
Thank you, ma'am, for your motivational and encouraging words, which inspired us a lot. Now, I request our co-convener, Dr. Richa Tandon, to conduct the program further. Ma'am, kindly join us.
Okay. A very good evening to all of you. I, Dr. Richard Tandon, Assistant Professor in Department of Botany, SS Khanna Girls Degree College, pleased to take this opportunity to welcome our distinguished speakers, our Principal Ma'am, Professor Lalima Singh, teachers and all the participants. Thank you, ma'am, for your blessings. Now, I welcome our first speaker, Dr. Ravi Tandon, who is Assistant Professor at the School of Biotechnology, JNU. Dr. Ravi Tandon did his PhD from University of Zurich, Switzerland in virology. Then he moved to San Francisco, USA to finish his postdoc research at University of California. He also served as staff scientist at Hawaii Center for AIDS, University of Hawaii, before joining JNU as faculty. He has published more than 33 research papers in high-impact peer-reviewed journals. His area of research is HIV immunology, virology, and infectious disease biology. He has obtained Young Scientist Award from the Young Scientist Award from the Association of Microbiologists of India and holds numerous research grants from various funding agencies. So I request Dr. Tandon to educate us on current COVID-19 scenario. Welcome, Dr. Ravi Tandon, for giving a.
So thank you so much for kind introduction, and I'd like to thank first everyone in organizing such a wonderful webinar, national webinar. And without taking time, I would like to share my slides. I hope I have authority to share my slides. So I'm still disabled. So anyone could kindly make me host. Okay, I'm host now. So I'm going to share it right now. I hope I'm audible to everyone. Am I audible to everyone? Can you hear me?
Yes.
Okay. So now I shared my screen.
Yes,
I hope you can see the presentation. So once again, thank you so much. So the topic that I will be covering in next 30 minutes, 25 to 30 minutes, it's very important because we all know that how COVID is influencing us, and there are many misconceptions as well, and many things people are not aware of how the COVID is going to affect people. So the title, as you see, today's talk is host response to SARS-CoV-2. So what we will be talking about, how we respond to the virus when virus goes inside our body. So before we discuss what kind of immunological response person generates once virus is in our body, we should little bit let's have a look at the background information of this SARS-CoV-2 or CoV-2 or novel coronavirus that we call. So it all started in December 12, 2019, where several clusters of pneumonia were reported in Wuhan, city of China. So what had happened, many people complained that they had some respiratory problem, and then they went to the hospital, and these people were found to be infected with a novel coronavirus that we call SARS-CoV-2 as well. So you see that it started in December 12, 2019, and till now we have more than 185. I mean, this number 185, I had written it down like a couple of weeks ago when I was preparing slides for some other event, but now it's more than 200 countries so far having people infected with the coronavirus or novel coronavirus. So when we say coronavirus, let's have a look what it is. So coronaviruses, basically, they belong to family Coronaviridae, and it's positive and single-stranded RNA viruses. They have different subgroups. So far we have 4 different subgroups of Coronaviridae family. We call alpha, beta, alpha, beta, gamma, and delta. And only members of alpha and beta subgroups, they infect mammals. And interestingly, when you look at all different kind of coronaviruses, so only 7 coronaviruses are known to infect human, while other coronavirus, they infect some other species. So what are these 7 different kind of coronaviruses? These 7 coronaviruses are like here. So first 4 coronavirus that you see which infect humans, they are human coronavirus NL63, human coronavirus 229E, human coronavirus OC43, human coronavirus called KU1. So all these coronaviruses, they cause mild common cold symptoms. Every now and then when season changes, we have complained that, we got cold. So we do not know whether it is common cold or whether you got normal coronavirus. All kids, it is school-going kids, they have runny nose. It is most probably they have normal mild coronavirus infection, which gets resolved in a couple of days. So they don't need to take, high, antibiotics or any kind of treatment. Antibiotic anyways won't work in case of viral infection. People just take it to avoid any secondary bacterial infection. So all these 4 coronaviruses are already known, and it's here with us since long. But in addition to these 4 coronaviruses, there are 3 more coronaviruses which causes SARS. SARS means severe acute respiratory syndrome. So the first SARS-CoV, yani SARS coronavirus, which caused 2002 pandemic, it started from China again. Later on, and this 2002 pandemic, SARS coronavirus, was not very aggressive. In a way, 8,000 people got infected around the world. Only 800 people died of this coronavirus infection. So the mortality rate was like high, with 10% compared to what we see here, like at 4%, 5%, sometimes even 2%. But there, it was not so contagious, because you could see 8,000 people got infected worldwide. That's why we didn't hear much about it. Now, another virus, which we call MERS coronavirus, which is known as Middle East Respiratory Syndrome. And this Middle East Respiratory Syndrome coronavirus, it caused 2012 pandemic. And later, what we see currently is called SARS coronavirus 2, SARS-CoV-2, responsible for 2019 pandemic.
Dr. Tandon, I'm not able to see your PPT. You're just in the document folder.
Okay, but I think there is some issue in sharing the slides. So you can't see my slides? So you can't see my slide? I think there was an issue.
Yeah. So all the time your document folder was visible. So you have to go to share.
Oh, surprising. No one. actually mention. Wait a second.
I was putting you a message, but you are busy in talk. Okay, good. So let's just, yeah, this folder was visible actually.
This can you see this now slide?
Not yet. The slide has to be shared. You are sharing the screen.
Okay. Okay. Wait a minute.
There is option to view what you want.
No, I understand. Wait a second.
Now it is clear.
You see the slides?
Yeah.
Now the slides are there. Before I proceed, I just want to make sure you can see. Abhishek, can you see the slide?
I am just seeing the same thing. Your folder I am seeing. So basically, if you are in a dual terminal, just take out the terminal card.
Now.
Yeah, now it's fine.
Now.
Okay, sorry for that, and I hope that everyone can see the slide now, or and you can hear me. And if any issues, please just intrude in between and just say so that because I'm not looking at my mobile while I'm presenting. Okay, so that's fine. Shall I proceed?
Go ahead.
Okay. So I was just mentioning that this pandemic, SARS coronavirus, started from December 12. And at this moment, there are more than 200 people who got infected with the severe acute respiratory syndrome, which is coronavirus 2. And it started from Hunan seafood market in Wuhan city of China, because all patients who had the symptom of pneumonia and who were also infected by coronavirus, they had a history of having visited in Wuhan city, see right here. And there is a Hunan seafood marker where bats were also actually sold. So people were actually linking the theory that it may have come from bats only. So number of countries so far is more than 200. And that's actually the present scenario. I also explained about the coronaviruses. They belong to family Coronaviridae. They are enveloped positive sense single-stranded RNA viruses, has been subgrouped under alpha, beta, gamma, or delta. Only two subgroup includes viruses that infects mammals. They are alpha and beta. And so far there are only seven coronaviruses, which we know that they infect humans. So what are these all 7 coronaviruses? I just mentioned for these 4 coronaviruses, NL63, 229E, OC43, and KU1. All these, wait a second. Hello? In addition to that, there are 3 more coronaviruses that caused pandemic. That started in 2002, which was SARS coronavirus, then MERS coronavirus, and then finally what we have, it's 2019 pandemic, which is SARS severe acute respiratory syndrome CoV-2. So this is actually the problem which we are facing now. And when we talk about the origin of this coronavirus 2, SARS-CoV-2, so we know that all these coronaviruses, they are originated from bat. So the bat has coronavirus, which has similarity of approximately 96%. And when you have similarity of 96%, you believe that the virus has originated from bat itself. But in general, you see that human and bats, they live in a different habitat. Since they live in a different habitat, then it is quite possible that direct transmission of this virus could not take place from bat. It may have involved some intermediate host. So for example, the original pandemic, SARS coronavirus pandemic that took place, it involved civet as a intermediate host. And another pandemic which took place, MERS coronavirus, which we call MERS, Middle East Respiratory Syndrome coronavirus, it involved intermediate host that was camel. For this novel coronavirus, which we call SARS-CoV-2, the intermediate host is pangolin. It's believed to be pangolin because the virus that we get in pangolin, it has a 94% similarity to that of the one which we have, this novel coronavirus. This is a delicacy in China. People also use it for eating, also natural bad. So it's still the question mark that you see that people are still making it sure, because there is a hypothesis, there is a chances that the virus came from pangolin. But this is how the origin of virus started. As far as transmission is concerned, so we all know that it transmits from by human to human contact. So one person sneezes or coughs in front of other person, so virus could actually transmit or affect or infect other person via droplets. Another possible way is through the droplet that falls over the surface of objects like a telephone receiver, doorbell, knobs, and immediately other person comes and touches it, and the person can be again infected by this route. So we all know about this basic thing. Now, what happens after SARS coronavirus transmission? This is very important. When we get infected, we do not know what is going to happen. That scares us a lot. First thing we should know that this is a self-limiting infection. When you say it is self-limiting infection, it means that when a person is infected by this virus, the symptom and recovery happens within 2 or 3 weeks of onset of infection. So most of the people with the are with mild symptoms and they get recovered very soon. In addition to that, when we try to categorize the outcomes of novel coronavirus infection, we have asymptomatic cases, which are 1.2%. We have mild cases 80.9%, severe cases 13.8%, critical cases 4.7%, death occurs in 2.3% of cases. Interestingly, it is also very interesting to note that the proportion of asymptomatic infection in children is much higher compared to that of adults. And when children get infection, the infection in general is less frequent, and whenever it is happening, it is very, very less severe compared to adults. So these are important feature about the transmission thing. Now, when we move on and when we look at the symptoms, what we know that most of the cases, you see the 98% of people with the novel coronavirus infection, they show fever. And 76% approximately people have cough, and 44% people, they also have fatigue. In addition to all these things, less common symptoms you find is the sputum production, headache, hemoptysis, and diarrhea. So all these complications that you see in coronavirus infection is well known. Now, when we go deep into the pathogenesis of this novel coronavirus infection, then we see that there are two different kinds of pathological subsets. These two pathological subsets, they eventually overlap with each other and they decide the pathogenesis of virus or the severity of disease. What are these two pathological subsets? One is virus response as well itself. So virus response, when virus goes in our body, what it does. So viral response is a one pathologic subset. Another subset is driven by post-inflammatory response. So you see that two things actually work together. Viral response initially and later host inflammatory response play an important role. Based on these two different pathologic subsets, there are different stages of coronavirus infections have been categorized. Stage 1, which is the early phase. Stage 2 is a pulmonary phase. Third one is stage 3, which is a very dangerous one, which we call hyperinflammation phase. When we look at stage 2, which is pulmonary phase, Sorry, just a minute. Actually, I think there is an issue with the host. I am just making someone else as a co-host so that they have authority to let people in. So I'm just looking at how to actually make other people as a host. Wait a second. So these three different kind of stages I was discussing. So stage two is important. What is stage two? Stage two has been categorized into stage 2A pulmonary phase, stage 2B. In these two, what we see that stage A is the one which is without hypoxia. The person do not have hypoxic condition. Stage 2B is with hypoxia. What is the difference between these two things? It's very interesting. I will explain one by one. Stage one, stage two, stage three, and stage one, what is happening? There is an early establishment of disease where the virus goes and it infects pulmonary epithelial cells. It's attached to a receptor which we call ACE2 receptor, and it has some co-receptor which we call TPP-RSS. With the help of co-receptor, the virus goes inside the cell, and then there is a viral ramp-up phase. An incubation period involves mild symptoms and non-specific symptoms such as malaise, fever, and write-off. Right after stage one, what we have some kind of manifestation related to our respiratory tract. So there is an involvement of a pulmonary tract, and once you have involvement of pulmonary tract, then we start to actually show some symptoms. Viral multiplication takes place and there is a localized lung inflammation. Very important part here is the low lymphocyte count, which we call lymphocytopenia. So our lymphocyte number percentage varies from 20 to 40%. And when it drops below 20%, then there is a problem. So low lymphocyte count is a very, very important or critical thing which we have to understand how it affects the disease pathogenesis. So viral pneumonia with fever is very common. Now here is the catch. When we talk about stage 2, we have one sub A group which we call without hypoxia, another one is B with hypoxia. What does it mean? So when we say it is with hypoxia, it means there is a lack of oxygen. How it is recorded? One, there is a ratio of a partial atrial oxygen divided by fraction of inspired oxygen, which is below 300 mm of Hg, then person is known to be hypoxic. So there is a lack of oxygen, and then there is a serious condition, because at this stage, patient may require hospitalization for close observation. And this hypoxic condition, where partial atrial oxygen fraction of inspired oxygen goes below 300 mm of Hg, at this particular time, patient may likely to go for ventilation, and this being on ventilation, it's not so nice, and recovery rate becomes very, very poor. And now it is also interesting to know the use of particular steroid drugs. So corticosteroids are being talked too much about, like people who have this infection should have corticosteroid, but it also depends at which stage you should be using corticosteroid. If a person is without hypoxia, so corticosteroid will not work, because corticosteroid will not let your immune system to develop. And when it is not developed, then what will be the problem? Then you will end up in very difficult scenario, very bad phase of coronavirus infection. But later, when a person actually continue or progress in the disease and develop hypoxia, then corticosteroid should be given, because at this stage you have to slow down your immune system, otherwise they will completely mess with your organ system and all that. So stage 3 is a very complicated, very serious stage, which we also call hyper inflammation, hyperinflammation stage. And this hyperinflammation stage is develops in minority of COVID-19 patients, because nowadays people are talking who should go to the hospital because beds are limited and government is not able to handle so many people who just visit hospital without any complicated symptoms. It is only for those people who get into severe stages, which we call hyperinflammation stages, which is happening only in minority of COVID-19 patients. How it is characterized? It is characterized by elevated level of inflammatory marker, which is IL-6, D-dimer, and several other inflammatory cytokines. At this stage, we have high neutrophil to lymphocyte ratio, which we also call NLR. Lymphopenia is another characteristic feature. The people who got admitted to ICU, then their CBC count was monitored, and it was found that most of the people, 82% of people have low lymphocyte count, which is 82% lymphopenia. So it seems that lymphocyte counts are very crucial. As you see in this figure, it tells us about the dynamic changes in lymphocyte and in COVID-19 patients. You see there are three categories of patients. The blue line here indicates those patients who had very low level of lymphocyte count right from the beginning. And you see that this blue line also indicates those COVID-19 patients who died. It means that low lymphocyte count is responsible for death. This black line indicates those people who had low lymphocyte count in the very beginning, but they preserved their lymphocyte count later. And when they preserved their lymphocyte count later, finally they got cured of COVID, despite that they had a severe infection. They had a severe complication during coronavirus infection. This orange line is pretty good because these are group of patients who had their lymphocyte count at normal range right from the beginning. That's why these people survived, moderately cured. It means that they had moderate or mild infection, and they survived the COVID-19 disease. So there is, how this lymphocyte count plays an important role. And when you see another interesting observation, lymphopenia predicts COVID-19 severity. So in this situation, the patients were grouped into two subgroups. So patients who had COVID-19 after day 10 to 12, their lymphocyte count was monitored. And those patients who had lymphocyte percentage above 20%, they were put under moderate group, and these patients got cured without any complication. While the other group, which had less than 20% of the lymphocyte count, they were grouped under pre-severe categories. And these people under pre-severe categories were retested after 7 days. So day 10 and then day 17. And at this particular point, they were again subcategorized into 3 different subcategories. Those patients who managed to get their lymphocyte counts above 20%, they got cured of infection. So it means more than 20%, they got cured. Another category is a severe one, where the lymphocyte count remained below 20% as it was previous time, but it was higher than 5%. So these group we call curable. Some severe complications were there, but the problem happens in those patients who eventually had lymphocyte count or lymphocyte percentage below 5%. They are few, and finally they also die. So our lymphocyte count is a very important predictive marker. So lymphopenia predicts COVID-19 severity in the very beginning. If you go and do your CBC, your lymphocyte count is about 20%. So it is most likely that you get cured of COVID-19 without any further complication. As you see that it is coming down, then there may be a problem, and then you have to see something. Here you see that there are different parameters here in viremia phase, acute phase, and severe phase. There are 2 components, which we call here the inflammatory marker, and the second one is D-dimer. These are inflammatory components. They keep rising when we proceed with the disease. And this red one is a viremia, and the viral nucleic acid, it also keeps going high when we go from viremic phase to acute phase or severe phase. This red one and green one, these are 2 components which represents a lymphocyte. They are declining as we are proceeding with the disease. So this is a complicated thing. So And lymphocyte count, drop in lymphocyte count is a dangerous thing. So having said that, we want to know that how a person with the coronavirus infection eventually dies. So how SARS-CoV-2 kills at severe stage, when you are at severe stage. You see on the left side here, this round structure, it is alveolar sac of a person, alveolar sac that we have inside our lung. So on the left side, it's an early stage, we don't see anything, but on the right side, you see that when a person is infected, so a lot of immune cells, they are migrating to lungs, basically in alveolar sac, and these pro-inflammatory cytokines, or basically all these immune cells, they start releasing pro-inflammatory cytokines. When they start releasing pro-inflammatory cytokines, so there is an enzyme called hyaluronan synthase 2. Here it is written HAS2, together with cytokine storm or inflammatory cytokine, they form hyaluronic acid, which we also call hyaluronan. So this hyaluronic acid or hyaluronan, what it does, it has ability to absorb liquid or water around 1,000 times of its molecular weight. So you can imagine how much water can it absorb. When they start absorbing water, the water is filled, like here, protein-rich filled water. When water is filled up here, it exerts pressure inside your alveolar sac, and this pressure doesn't let oxygen or CO2 to exchange between arterial blood or alveolar sac. So it restricts gaseous exchange, as a result of which you are not able to breathe properly. At this stage, people need ventilation, and that is a problem. Another thing is that there is a feature called a disseminated intravascular coagulation, which is characterized by systemic coagulation of blood in different organs. So this is a characteristic feature of all those non-survivors, because postmortem was performed and they said most 71% people who died of coronavirus had DIC. So to tackle that, people are given low molecular weight heparin, LWMH, which actually dissolve or kill the clot. And this is used for very severe cases so that they could live even at the severe cases. Now, these are actually disease pathogenesis, which I mentioned. It means that lymphocyte is very, very important and we have to maintain it. But actually, what are the current treatment options we should be knowing? The drug repurposing people are actually are focusing on, because having a new molecule for the treatment of coronavirus infection might take a long time. So hydroxychloroquine, which was originally used for treating malaria, is now being considered to treat coronavirus infection. What it does basically, it actually restrict fusion of lysosome with that of endosome. So when virus goes inside cells, it forms endosome. There is a fusion with the lysosome. This fusion, hydroxychloroquine, stops, and as a result of which the pH inside lysosome doesn't become acidic and remains alkaline, as a result of which the final cycle, full cycle of virus doesn't actually take place. Another drug is called remdesivir. Remdesivir was originally developed for treating Ebola virus, but it didn't work. And now they are actually using it. Clinical trials are happening for treating COVID-19. What it does, it is a nucleoside analog, while replication process instead of adenosine, this remdesivir comes, and as a result of which the replication or transcription of nucleic acid of the virus doesn't take place properly, and then you break the cycle of the virus. Some protease inhibitor which were used for HIV treatment, lopinavir, ritonavir, these protease inhibitors were also used for the treatment of coronavirus. And as you must be knowing that in Jaipur, first two persons got cured of COVID-19. They were given lopinavir, ritonavir together with hydroxychloroquine. As I was actually mentioning early, that virus enters inside cells, lung cells, or pneumocytes, or epithelial cells of pulmonary sac or respiratory epithelial cells. So these are pneumocytes, and this is ACE2 receptor, the important one. Virus recognizes this receptor and goes inside the cells, but it is not enough. It also requires a co-receptor, which we call TPP-RSS2. With the help of TPP-RSS2, this virus goes inside pneumocytes or lung epithelial cells. So it means that if TPP-RSS2 is not there, then it will not go inside. So another drug which we call serine protease inhibitor, it's called camostat mesylate. This camostat mesylate inhibits TPP-RSS2, as a result of which virus cannot go inside the cell. This is also a potential therapy. Nafamostat is another TPP-RSS2 or what we call serine protease inhibitor, which is currently used. Another approach is that we can use soluble receptor. So receptor that is present on the surface of cells cannot be realized directly. So there are soluble receptors. These soluble receptor will wash out all the viruses, and as a result of which it will not bind to the cells, and this way the person will be protected. This is under clinical phase 2 trial. Antibody, monoclonal antibody against receptor binding domain of SARS coronavirus 2 is also being considered for the potential treatment. It is also under clinical trial. And plasma therapy, of course, you have been hearing in the news and other places where people were given plasma for those people who got cured of infection, and but there is a variability because some people develop very poor level of neutralizing antibody, so they may not confer the protection if the plasma of those person is given to the sick people. So it is very important that we should titer the plasma before we use it for the therapy. IL-6, which causes massive cytokine storm, which also is responsible for augmenting this hyaluronic hyaline membrane in the alveolar sac. People use antibody against anti-IL-6 receptor, which blocks it, and as a result of which IL-6 doesn't execute, is a complicated or complication. It is called tocilizumab. It is IL-6 receptor antibody. This is a very important one. I think I'm just finishing it. It's almost last slide. This is called hyaluronidase. Hyaluronidase is the one which dissolves hyaluronan or hyaluronic acid, because I said that when the water is filled in alveolar sac, there is a problem in gaseous exchange. But if you use hyaluronidase or HAF2 inhibitor, it will eventually clear this hyaline membrane, which is caused because of hyaluronan, as a result of which the gaseous exchange could be initiated later and the person can get cured. Vaccine is a long way, and don't know when it will come, but to have alternative approach, people are using the one which is already available. So BCG vaccine has been found to reduce yellow fever virus or viremia by 71%, and Mengo virus is another virus where BCG vaccination plays a proactive role. People are trying and seeing whether BCG vaccination could help in conferring protection against coronavirus infection. Another thing is that CSIR is also testing anti-leprosy vaccine, whether there may be a cross protection, eventually we can get cured of, or we can protect ourselves from coronavirus infection. And UK people are trying MMR vaccine, which is given for rubella, mumps, and measles. People believe that in this vaccine there will be a cross protection, or it may confer protection against SARS-CoV-2 as well, because of cross immunity or cross protection. So then conclusion is that outcome of COVID-19 is highly variable, and the spectrum of available antiviral or anti-inflammatory drugs depends on the stage and severity of COVID-19 drugs, when drug is administered. So for example, there are 3 stages of infection. At which stage you should be using which therapy, this is very important. Staging of disease is very important, and then only we will be able to save the lives of people who are at critical stages. So drug repurposing as of now appears to be the quickest way to mitigate the complication of a stage, because we do not know when actual drug for SARS coronavirus will be in the market. So in the long run, no doubt, vaccine seems to be the only way to get rid of this SARS-CoV-2 infection or COVID-19 problem. Till then, what we can do, we can just keep ourselves away from being in a crowd, follow social distancing, use mask, don't shake hands. Namaste is the only way. So with these things, I'll stop my presentation and I will be happy to take any question you guys have. Thank you so much.
Thank you. Thank you, Dr. Tandon. Thank you, Dr. Tandon, for your excellent talk, giving comprehensive overview on COVID-19 pathogenesis, which I believe is very important to understand in order to develop potential therapy for COVID-19. Thank you once again. Now, we invite our second speaker, Dr. Abhishek Kumar, who is a faculty research scientist at the Institute of Bioinformatics, Bangalore. He is a recipient of prestigious Ramalinga Swamy Re-Entry Fellowship awarded by Department of Biotechnology, Government of India. Dr. Kumar is working on development of translational multi-omics approaches for studying various human diseases like cancers and neurological disorders. Moreover, he also started genome-wide characterization of coronavirus proteins and their motifs for potential therapy. Welcome, sir. Welcome, Dr. Abhishek Kumar, for delivering a lecture.
Thank you, Dr. Richaji, and thank you, Dhanyavad, Principal Mahodaya and Organizing Committee College. And it's my pleasure to come somehow or other to Allahabad. I was my BSc was from Allahabad, and it's a wonderful city to be. And let me start my presentation.
I made you co-host as well, because I was co-host. Now you are co-host.
Yeah, sure. So let me start the presentation. And okay, so it gives me a lot of choices to choose from. So I'm just checking whether I'm correctly connecting correct PPT here. So just a minute, yeah. Let me see if my PPT is on.
Dr. Richard Edward Madam, shall we have questionnaire session?
Just 1 minute, yeah? Just a minute. 13. Okay, yeah, I'm in the correct one. So let's go.
There are few students who have put their question in the chat room, so I'm also answering them, because some of them they sent me question privately, so I am also trying to answer them in the meantime.
Yeah, sure. You do that, then in between I'm keeping my PPT ready. Yeah. So this is the PPT. Okay, so let's me have. Yeah, so you can answer them maybe. Just a minute. Yeah, so let's get yeah. Yeah, so I'm ready. Can you see that?
Yeah.
Okay. So everybody is seeing a curtain. So that I am.
Yes, we have seen.
So thank you again. Thank you to the organizer for invitation, and I'm going to talk about how the viral genomics is working during pandemic of COVID-19. And Already Dr. Ravi Tandon has told that a wonderful overview on COVID-19 and SARS-CoV-2. So I'm going to discuss. So basically, I'm a bioinformatician or a genome biologist, and so we had several core areas where we were working. But when the pandemic struck, we also faced challenges, and the challenges put us into the let's challenge virus in some way, and that's when we started working with viruses. So as all of you are aware, as a student, and even if you're not aware, you'll be aware that viruses are an considered to be a living and in between a living and non-living entity, because it has a genetic material and some proteins, but it do not have self-propagatory lifestyle. Means it requires a host to propagate. And there are several viruses, and there can be very big viruses like Ebola virus, and there can be very small virus as porcovirus. And so SARS-CoV-2 or also HIV are in the mid-range virus with size of 120 nanometers. And so you have an idea how different viruses look like. So this is a wonderful figure organized at the viral zone at Expassi. And with what happened with the SARS coronavirus, which all of you are aware, that it originated from a Wuhan wildlife and seafood market. It's like a machli market, but other than machli, they're selling a hell lot of other organisms, and these are wild animals. So in Chinese ancestry, the going wild and catching animal and eating was a predominant, obviously everywhere, but in China it's considered to be very luxurious, and it's a business of rich people today. And therefore, as China become richer and richer, people started getting these animal from the wild from wet market, and Wuhan is only one example. And plus so this becomes so famous in society that anteaters are actually exported from India, China, and many other countries, and many other animals actually. So basically the Chinese or Asian ones are finished, so they are getting it from another continent by illegal smuggling. So these were received. We'll answer the questions later, and this is one example of Wuhan market where you see the different wild animal life. So in a wildlife, the life is very different, and viruses and other organism dynamics is quite different, and that comes directly to the market, and then we get infected. So if you are aware of the coronaviruses are of various kind, and they are, as Dr. Ravi Tandon told, they are grouped into 4 groups: alpha, beta, gamma, delta. And it is known that alpha viruses also infect human from bat to intermediate host, and intermediate host to a mammal, primarily a mammal, and from a mammal which human in interacting will get it. So you'll get cold. Normal cold viruses are listed, and beta viruses are also same. But then came emerging disorders like SARS and MERS, and now the pandemic of the SARS-CoV-2. And these are the most dangerous viruses. If the MERS is even much more killer. It's a killer virus, but it's not spread as SARS-CoV-2, luckily. And then we are safe. So basically, what this slide is showing is that there are at least 2 groups of viruses, coronaviruses, that can infect human. And this is primarily coming because we are invading more and more wildlife, and we are picking up wild animals into our food, primarily in China. And so our encroachment to the wildlife has to be limited. And of course, if you are aware, in a classical Indian village or town you grow up, parents will say not to catch the wild cat or wild animal. Stop it, don't catch it, you may get sick. So, or maybe my grandmama used to tell me, to warn me for going wild, especially towards bats and cats. And this is clearly visible here. So, probably she was not knowing that about coronavirus, but she was aware that I will get sick. And that's how the concept is, that you have to keep a distance from wild animal, and from your natural habitat you must not start becoming wild. And our natural habitats are mostly town and village, not going into the wild. And that's when coronavirus came in. And so coronavirus, when they're infected, is genome also get published, and we a Chinese group has published in Host and Microbes journal, and that shows that this is about 30 kb genome with 14 genes and 27 proteins. And many of you who are students will see one gene, one protein hypothesis. What happens here? Unique that 14 genes and 27 peptides. So actually, there are 4 structural peptides, which are a spike protein, envelope protein, membrane protein, and nucleic acid protein. They are at 3' end, or 3 towards 3' UTR. And then the larger peptide, which is covering about 67% of genome, is producing a large peptide protein first, and this protein is broken into small pieces, and 15, 16 pieces, and so the overall you have 27 peptide. And these bottom slides are actually showing the evolution, and evolution means there are 4 types of viruses, so they will evolve into 4 different clusters. And that's primarily that's what is shown in the bottom part of the slide for each first for whole genome, and then different genes and proteins. So how the breakup is actually, you have ORF 7,000 amino acid, and it could be breaking up into non-structural peptides 1 to 180 and 181 to 818, and then so on, and it will break into different peptides. And these are the non-structured peptides which have different function, and some of them has no function, and some of them has like NSP5 is a polypeptide which has a inhibiting function. And then you have NSP14, which has a ribonuclease, exoribonuclease like function, and 14, 15, and so on. So they have whatever is a primary set of protein virus requires, it brings it has, and when it infecting it will help in the virus propagation. And other proteins will be utilized by the host system. Other proteins will be from they will utilize from the host. So let's go. And then already Dr. Tandon told you, and I'm also showing you that bat is the primary source, and from bat the intermediate host, primarily a mammal, will cause a disorder. And so this is anteater is one of the example for here, and then people may question that, but that's how most of the coronaviruses of current time reach to human. And so we were working on different aspects on coronavirus, whether we can utilize their proteins to a study. And one of these protein, nucleic acid protein, we have studied in detail, which is under consideration now. And this protein is actually this protein is actually can go to the nucleus, and also we are seeing some hint that it will be in the cellular localization. And then what we see here is that this genome is 30 kb genome, and that 30 kb genome is conserved across different bat coronaviruses, Indian samples, and the Chinese sample obviously is a reference sample. And towards 3' end is the is the nucleic acid protein, which is called N protein in short. And this protein is actually having non-structural proteins at the domain at the C terminal end and 5' N terminal end and C terminal end. And then this one will has the has N terminal domain, which is ribosomal binding, and the ribosomal binding is C terminal end, which has a dimerization function as well. And there is intermediate linking domain, linking region, which will link. And then during our study, we find that there are 3 nuclear localization signal, NLS, and 2 nuclear export signal. And other than that, there are 7 motifs. And that's what we are going to discuss now. And this is the protein sequence alignment. Top ones are the SARS coronaviruses, and the bottom ones are other viruses. And what we see here is a clear cut that SARS coronaviruses are much closer to each other, obviously, and then the other viruses of other classes are looking different. And but what we see here, that there is a motif 1, which is which is present in the in this region, which is the conserved motif. And then the second motif is present here. And then we keep on looking for other motifs. What we see that the third motif is repeating S and R residues, serine and arginine residues. So SR motif is found in this region, and which is known for these viruses already. And so this motif is present. And then we see the motif 4, which is not found in other than SARS coronaviruses, but it's not found. It's completely conserved here. And then what we also notice that there are 3 more motifs, plus there are repeating glutamine residues, and this is called, we have named it glutamine patches. So there is a repetitive one here, massively repetitive one, and then repeating again and again a conserved. So there's the different color shows a different pattern of conservation. So fully conserved ones are black, and one which is there's a one which is missing, got mutated in SARS-CoV-2. Primary reason is to see how the conservation and so then there was kindly. So okay, so and then we also identified the second patch, which is here, towards C terminal end. And along with these patches, nuclear localization signals were also conserved, and the motif 7. And then we have, we can go, there's a nuclear localization signal, 2 nuclear export signals also present. So what we saw that this is SARS-CoV-2 is actually very close to bat coronavirus, and it is 90% identical to SARS-CoV, first SARS-CoV, and but much nearly identical to the bat coronavirus. That indicates that indeed bat was provider to us. And then these are the 7 motifs we have identified. Apart from the motive, we have characterized those motifs with these motifs, how the localization of these motifs are. And then what we see that these motifs are actually, motif 2 is actually highly conserved, and it is indeed 100% conserved in this, if you count only these patches. And then these motifs are found in other viruses as well. So if we want to develop an artificial peptide, this can be a good peptide by which you can develop vaccine. And this by developing the vaccine, we will find a we'll find we can actually provide protection against all members of coronaviridae. And yeah, so that then basically even for mild fever and cough, it will be effective. And then but there could be cases when you need a more specific SARS-CoV specific motive. We have second motive, which is motive 4, and it is also nuclear export signal, and we have compared with other nuclear export signal in HIV and also in human. So this is slightly different. So this better, not slightly, for with human it is much more different. So therefore it can be very easily placed as a candidate for artificial peptide synthesis. And also what we saw that during our study, that these are this protein is a highly unstructured in region in nature. And unstructured means it will not fold properly. So within the foldable and unfoldable region, so these are the very high going graph, and it's going to us above 0.5 is showing that this is unstructured region. And between unstructured region and a structured region, there are 7 motives we have identified, and these structured and unstructured region are actually carrying the SR rich motive and a glutamine rich region, and which has a similar patterning in the other viruses with few exceptions. And these are the tools which we have used for the calculation of structured versus non-structured region. So when we look at the glutamine rich patches of nucleic acid protein, what we find that there are 20, in first patches is about 20 residues, and second patch is 16 residues long, and these are the residues which we are counting. And so this when we take those messages together, we'll pull those messages together. What happens in biology of the coronavirus, that nucleic acid protein have already known that postulated that it has a multiple function and multifaceted protein. And so what we said that this protein is actually has 3 nuclear localization signal, means it can bring bring itself and also associated peptide to the nucleus by binding the other peptides and come to the nucleus. Once the task is finished, it can bring them back, or it can come back to the cytoplasm and retain there by virtue of 2 nuclear export signals. Nuclear export signal will bring it back to the cytoplasm, and then it will stay there. And this motif 4 and two Q patches will aggregate several copies of this protein, and then that will cause aggregation. So if you are already seeing that SARS-CoV is aggregating in different cells, that could be a hint. And we have ourselves not performed the experiments, but we have found that SARS-CoV and some other viruses also have this tendency. So and the signals, and both dynamic transporter function and aggregate function is potentially a regulatory mechanism of different viruses, which is not previously well depicted. But there's some examples, and viruses are so many that we tend to forget one. But when we carefully looked into the literature, we have found. So with this, this is the overview of this nucleic acid protein. And with this, I'm indeed coming close to end of my slides. And So definitely we are talking about funding agency in institutions and also to whom we are thanking, and to detail that I have this coronavirus volunteers and my team in the bottom, but when the coronavirus came, we had to set up a new team for volunteers who can work from home during pandemic, and they all are actually working on different projects, and indeed we have a good amount of data, and which will be public soon. And there's some postdocs, there are some PhD students, some master students, and even some of them are bachelor students. So we thank them as well as our funding sources, and we thank you. And if somebody want to become volunteer or want to know more about our group, they have email ID, mobile phone number, and the website is given. So please contact us if you want to be part of this team. And yes, so at the same time we are actually also announcing that we are accepting in International Journal of Computational Biology, SARS-CoV-2 papers are being accepted, and so if you are interested, please submit there. And with this, I'll thank you.
Coincident. So this is a flavanol, as I said, and it is, if we want to consume it, we can consume in the form of red onions or the white onions or pears and spinach. So this quercetin actually inhibits the oxidation of LDL, that is the low density lipoprotein. That's the bad cholesterol, and hence prevents the cardiovascular diseases. And it's also found to have some effect on the cancer and also has an antiviral action. It has antihistamine effect and also improves the inflammation of the prostate gland. So it is wise to consume a lot of hot peppers, red onions, white onions, pears, and spinach.
Sorry, I think there was some disturbance. I had to unmute. I had to mute all as a host. So anyone who would like to join in, please unmute yourself.
Yeah, so we are open for the questions. Yeah. So let me see if there is a question.
Please unmute.
In chat box, I have given some questions. So please read out the questions.
Yeah. So
this question to Professor Tandon and one more question to Dr. Abhishek Kumar, please.
So there are a lot of questions actually coming in. So one question Dr. Selvaraj has asked whether there are some comfortable article on COVID-19 has originated from Wuhan. Actually, yes. The Holmes, who is an Australian researcher and very big virologist. So he's sending. He has the paper. They actually went to this market, and the picture which you saw in my presentation is from. They took it from the market actually, and so they were already knowing that this market is a permanent source of virus, and obviously there can be argument and disargument, and that's how the world is moving. But they have a push that this market is the primary source. And then we have a question that how mechanical vibrators play a role in inactivating viruses. So Dr. Ravi London, can you answer this question?
Sorry. I've been answering on this open chat room. So which specific questions you are referring to? Because there are so many.
There was a question on the mechanical vibrator.
Mechanical vibrator. Well, anything. Well, you see, I haven't seen anything which is like under clinical trial as of now. But if someone has identified it, it has to go through several clinical trial or several other, research before you could actually use it as a, normal therapy or any kind of thing. I don't think anything like this is open yet, or people are doing clinical trial on that. Because I
Yeah.
am not able to, unless until you were talking about a particular radiation which is specifically killing it, or there are certain nanoparticle based therapy where you get a probe linked with the nanoparticle, and then they actually have certain marker which actually goes and bind to those specific cells which are infected. But at this moment we are in very early stage of research on SARS-CoV-2, because we do not know a specific unique markers which are expressed in infected SARS-CoV-2 cells. Once we know that a particular infected cell is expressing a unique receptor, you could use this nanoparticle based strategy to target it or kill it. So I think this will be very naive as of now to comment on any vibrator based mode of the therapy or anything.
Thank you, sir. Thank you.
Anyone has any question could just come on mic and just ask us. I have been reading, I believe, all the questions. If it is referring me, I have already typed, and if someone didn't understand, I am happy to answer it right now here.
So I'm sending the question paper required by doctors. Doctor, so just a minute, I'm sending it host, and it was published in Host and Micro. So so
Hello.
Hello.
Am I audible?
Yes.
There is some issue on internet. Thank you. Thank you once again for Dr. Tandon and Dr. Abhishek Kumar for your valuable information about COVID-19. The genome-wide characterization and computational analysis of coronavirus protein will certainly help us to find potential therapeutic target, and I believe during this pandemic, bioinformatic analysis is the only way to proceed further while all the labs are non-functional. Thank you once again.
Thank you so much. I am also encouraging students who are linked with us, this online platform, to ask any question if they have. They have been typing, and I have been trying to respond each and every student effectively. But still, anyone is there who would like to say something in real-time manner, it is easier for me because typing takes time.
yeah, so I have sent the paper in the public so that they can read, and also to Dr. Selvaraj. So yeah, so thank you again for inviting.
Thank you. And at the end of this session, I would like to thank our convener, Dr. Preeti Singh, members of the organizing committee, all the teachers, research scholars, and the participants. Thank you.
Thank you. Of anyone, any student who is associated right now is not recalling any question, or is hesitating in asking any question as of now, feel free to shoot us an email anytime they want. We will respond. So yeah, so good luck everyone, and thank you so much for giving me this opportunity because it is important that what we are doing, what we are studying, we should convey it to students, because these are the people who would need to understand. They are the people who would pursue their career in research and find the ways which kind of research, what kind of research they should be doing. So they should be excited at this moment, because they are the people who would eventually lead this academic thing, further and do research on this. Okay. Okay, so we end this. Okay, thank you so much.
Thank you so
everyone.
much.
Thank you so much.
Okay.